Welcome to the MCAT Basics Podcast, brought to you by the physicians at Med School Coach. Each week, Alex Starks breaks down high-yield MCAT topics, so you can score as high as possible on test day.
Hello, and welcome to Prospective Doctors Podcast, MCAT Basics. I'm your host, Alex Starks, and our goal for today is to master more metabolism, specifically the Krebs Cycle, also known as the Citric Acid Cycle and also known as the TCA, or tricarboxylic acid cycle. So this content is really important for one major reason. It shows up in both Chem Phys section as well as the Biochem section. This episode is also a continuation in our series on metabolism, and if you recall, in our previous Metabolism podcast, we discussed glycolysis. We focused on that metabolic process as, quote, the robbing of electrons from glucose in order to power an insatiable power factory called the mitochondria. Where we left off was focusing on the final product of glycolysis, this 3-carbon molecule called pyruvate. And I want to stress, again, the structure of this molecule being very important, so I'll just review it real quick. So carbon 1 is a carboxylate, so a COO-. Carbon 2 is a ketone, so a C double bond O. And then carbon 3 is just a boring carbon, has 3 hydrogens connected to it. So last episode, we also discussed how pyruvate's journey can continue either in the presence or absence of oxygen. So today, we're going to continue following the oxygen present or aerobic fate of pyruvate. So the first thing that happens is that it's going to enter into the mitochondria because glycolysis occurs in the cytosol. Now, pyruvate's entrance into the mitochondria is mediated by a complex. It's a complex of enzymes, 3 actually, called the pyruvate dehydrogenase complex, which I'll just call PDC for short. So this step is very unlikely to show up on the MCAT, and I can think of one and only one AAMC practice question that requires knowledge of the steps. And I don't know about you, but I would be happy to take the L on that one so that I can fit more and higher yield facts into my brain. So just for this next part, keep that in mind. For the sake of completeness, here's what I think you need to know. So the PDC is this 3-enzyme complex that converts cytosolic pyruvate into mitochondrial acetyl-coa. So not only are we moving from the cytosol into the mitochondria, we're also chemically modifying our substrate. So this involves a decarboxylation of pyruvate, which generates carbon dioxide. And there's also generation of NADH, which then goes and deposits those electrons into the electron transport chain. Another potentially useful fact here is being aware that there are a bunch of cofactors that are involved here. One of which is called lipoate, and this is the conjugate base of a fatty acid, lipoic acid. And lipoate helps to move the substrate from one complex to another. When I was in biochemistry, I remember my professor describing it as it was like this arm, or this crane almost. And pyruvate's attached to this crane called lipoate, and it's shuttled between these different complexes. So maybe that helps. But the main thing here is that pyruvate goes into this factory called the PDC, and out pops acetyl-coa, ready to join the Krebs cycle. Now, as a side note that we'll talk about in a later episode, the acetyl-coa here can be used as a building block for fatty acids, but today we're only going to focus on the catabolic fate. Now that we have acetyl-coa in the mitochondria, specifically in the mitochondrial matrix, we can subject it to further oxidation, and we can strip away even more electrons to power the mitochondria, using NAD plus and FAD as the shuttles to move the electrons from the substrate into the electron transport chain. One quick note, something I didn't mention earlier, with the names of this cycle, the citric acid Krebs cycle and the TCA or tricarboxylic acid, I interchange them normally and regularly. I don't have a favorite, so if I say one of them, I mean them all as a collective. Now, before we get into the actual process, the steps of the TCA cycle, I want to emphasize something important about visualizing what these molecules actually look like. And understanding how the structure of these molecules are being changed and manipulated is going to be essential for deeply understanding the importance of every metabolic step here. For starters, let's take a moment to make sure we know what acetyl-coa looks like. We discussed that a 3-carbon pyruvate is decarboxylated in that PDC, that whole process, meaning the acetyl component, being a 2-carbon molecule, is specifically an aldehyde. because we cleaved off the carboxylic acid portion of pyruvate, leaving just what used to be that ketone of pyruvate. But of course, when you're terminal carbonyl, you're going to be an aldehyde. All right. So the coenzyme A molecule that's attached to that carbonyl carbon of the acetyl molecule is not an important structure. If knowledge of the structure is ever required, you'll be given the image. They'll make you reason about the chemical properties of its functional group, something like that, but the actual molecule is not important. Now, a quick study note on how to approach this type of material, similar to advice I would give my students about like memorizing the amino acids and whatnot, is to attach some like weird or cool or unique thing to the step or the molecule so that it helps to cement all of the other useful and testable facts that you might need. And I'm going to try and point those out, or at least ones that I use. There are other ones out there, so if a cool fact that I throw in doesn't help you, just go find something different. Alrighty, step 1. Acetyl-coa is added to oxaloacetate to make citrate. So in this step, what we're doing is we are adding the acetyl group, that 2-carbon group that used to be pyruvate, to the substrate formed in the previous cycle. So when the TCA cycle was done, we have an oxaloacetate molecule that's ready to become chemically attached to the acetyl portion of acetyl-CoA, and that it produces citrate. So this is an irreversible reaction, and it's carried out by an enzyme called citrate synthase, which creates a new carbon-carbon bond between oxaloacetate and acetyl-CoA. Now, this is powered by breaking a super high energy thioester bond of acetyl-CoA. That's what actually attaches the acetyl to the coenzyme A with water. It's a hydrolysis there. So this resulting chunky 6-carbon molecule is like glucose. It's going to be torn apart for its electrons, and it's going to give off carbon dioxide molecules until we get back to that 4-carbon oxaloacetate at the end of this cycle. So one notable special fact about this is that this whole mechanism here is simply a Claisen condensation, which I won't explain today, but is an organic chemistry mechanism that you might need to know. But the fact is kind of boring in my opinion, so I'm going to give you something that's a little bit better, I think. So imagine an ester. An ester is going to be a carbonyl carbon that is also bonded to an oxygen. That's that functional group. So a thioester is just that same carbonyl carbon that is bonded to a sulfur instead. So that bond is really high energy. It's actually, depending on where you look, a little bit more or a little bit less than the hydrolysis of ATP. Somewhere around that negative 31, 33, 30, depending on who you ask, kilojoules per mole. And so that is a lot of energy. So we're gonna come back to that though. So this is one of those interesting thioester cleavage steps. Alrighty, so we have citrate. Now, citrate is going to become isocitrate. Now, our catabolic machinery doesn't like the shape of citrate. In particular, it doesn't like that there's a hydroxyl group on carbon 3. And what do you do when you don't like how your room looks, or a molecule looks? You rearrange it. So the cell does this by using an enzyme called a aconitase that first removes the hydroxyl group from carbon 3, because that's not where it wants it, and that hydroxyl group loses water, and then it adds it back in, but this time at carbon 4. This might seem a little extra, but it is because the original positions of citrate's hydroxyl groups, they're not conducive to the oxidative decarboxylation step that we're going to see in the next step. So it's like a setup step. Oh, and the cool thing I think about this step is that we are taking citrate, which is actually a symmetrical molecule, and it's achiral. There's no chiral carbons. And if you don't believe me, look it up and try and find a chiral carbon. And we turn it into something that is chiral. So that's why we call it isocitrate. So our next step is uber important. You have to know this one. So isocitrate becomes alpha-ketoglutarate. All right, so now that we have our isocitrate, we're ready for a oxidative decarboxylation step. So this occurs in 2 steps. two different reactions. We have an oxidation, then we have the decarboxylation. And they're both carried out by this enzyme called isocitrate dehydrogenase. All right, so isocitrate is oxidized, or in our narrative, it's being robbed of its electrons. And those electrons are placed onto NAD+ to form NADH, which can then go deposit the electrons into the electron transport chain. And that intermediate that results from that reaction is then decarboxylated, and we lose a CO2, or carbon dioxide molecule. The result's alpha ketoglutarate, and if you're following along with like structures, make sure to note that this molecule is 5 carbons long with a carboxylic acid at both ends. And then the alpha part is that we have a ketone at the alpha carbon. All right, so the presence of that ketone at the alpha carbon actually helps to promote a second decarboxylation event. Now, a fun fact here, and actually, I'll come back to this fact when we talk about fatty acid metabolism, but when you're exercising, the CO2 that you're breathing off, heck, even when you're just sitting here listening to me, it does come, for the most part, from the carbon dioxide lost here from the TCA cycle. So whenever you go to the gym next time that you're doing something physically active and it gets your breath going, try and remember, hey, I am decarboxylating isocitrate and I'm getting alpha ketoglutarate. Alrighty, our next step, alpha ketoglutarate becomes succinyl-CoA. So we have this unstable alpha ketoglutarate, and we're ready for our next oxidative decarboxylation. Now, if it helps you to think of like, oh, alpha ketoglutarate is unstable, why is that the case? I want you to draw the molecule out, and like I just described, we have a carboxylic acid at a terminal, and the alpha carbon is a ketone. That's a lot of oxygen, all sort of concentrated in one area. So draw it out, see where the electrons are, and it might help elucidate why it would be unstable. Now, just like the previous step, alpha ketoglutarate is going to be oxidized. It's going to give its electrons to NAD+, makes it become NADH, and we also get rid of a carbon dioxide molecule. This step is a little bit more complicated because of the acetyl-CoA being added to alpha ketoglutarate to make succinyl-CoA. And how I want you to kind of remember this, I think it'll be helpful for your test, is this is a complicated step because of the CoA, and so we need a complex of enzymes. Very, very similar to how we took pyruvate, we threw it into the PDC and out popped acetyl-CoA. So very, 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 very similar. But the major takeaway, overall, we have a 5-carbon molecule in alpha ketoglutarate, and we decarboxylate it. We steal a little bit more of those electrons, and we get this 4-carbon succinyl-CoA. And notice here now that succinyl-CoA and our final substrate we mentioned, that oxaloacetate, both have 4 carbons. And so this is going to mark the very last step where we're going to lose a CO2. And the original acetyl-CoA is basically gone now because it only had 2 carbons, and we've decarboxylated both of them. I'm going to let that be the fun fact for this one because I'm having trouble thinking of anything more interesting. This is the step that we lose our 2nd carbon. Alrighty, our next step, which I believe is step 5, succinyl-CoA becomes succinate. So that means we're removing the CoA and we're getting this molecule called succinate. So you might be thinking, why did we even put the CoA back on after we cleaved it off? In step 1 from acetyl-CoA. The reason why is because the mitochondria in the cell is just a very greedy bastard. That's just sort of the way it goes. And this is a mechanism in order to get more ATP. So like that first step where we cleave that thioester bond in order to attach oxaloacetate and the acetyl or acetyl group from acetyl-CoA, we're going to be doing something very similar. We're going to cleave that CoA bond to succinyl, and that energy is actually going to be used to take a GDP and attach a phosphate to it. This makes GTP, and then something that I think is kind of neat is the cell has this enzyme, I'm not sure what it's called, but it has this enzyme that will just remove the guanine and add an adenine there. So we have ATP, and there's our energy. So let me just recap a couple things I want you to know about this step. Is 1, there aren't any phosphate groups on succinyl-CoA, so we have to add a free inorganic phosphate to GDP. So that's to make GTP, and that's something that is of note here. And also, the reaction's catalyzed by a synthetase. This is something that I've seen my students get wrong over and over and over and over again, and I got wrong when I was studying, is that a synthetase creating this GTP is not a synthase. A synthetase usually uses ATP to do something. It's a ligase, so to make some sort of covalent bond. But here, because of that thioester cleavage, we're able to send it in reverse and use the free energy from that thioester cleavage to have the enzyme essentially work backwards, if you will. So instead of cleaving an ATP to do something, it's actually storing energy and forming an ATP. Now, a synthase forms a bond without the direct use of ATP. So there is the distinction. And for anybody that has sort of previewed and tried to read ahead and look at the electron transport chain, you might be thinking, wait a minute, ATP synthase makes ATP. Like, what's going on there? That is going to be explained in depth when we talk about the electron transport chain in the next metabolism episode. All right, so the next step, succinate, becomes fumarate. Now, I'll just throw out there that all of these words I've heard different professors say differently, like succinate or succinyl-CoA or fumarate. So these are just the ways that I've heard it. So I just think that's a funny thing about biology. But anyways, I digress. Okay, so now we have succinate, which if you've been tracking the structural changes we've been making, it's got 4 carbons. Carbons 1 and 4 are carboxylic acids, and 2 or 3 are just hydrocarbons. Nothing fancy at all. So the issue with this structure is that it isn't the best electron donor, because we want to strip away these electrons, but the cell recognizes it can still shake our substrate down for a little bit more. Now, because succinate isn't that great of an electron donor, we need a carrier molecule or electron acceptor, if you will, that is more oxidized than NAD+. NAD+ just isn't strong enough to convince succinate to give up its electrons, but FAD is. This is all catalyzed by an enzyme called succinate dehydrogenase, and it takes these two molecules, succinate and FAD, and produces FADH2 and fumarate. So basically, the enzyme pulls hydrogen atoms, complete with their electrons, not protons, but hydrogens, off of carbons 2 and 3 of succinate, which again are just those boring old hydrocarbons, creating fumarate, which has the double bond between those 2 carbons. Now, the fun fact or memorable fact here is that, and it is very important for the MCAT actually, is knowing that this step occurs inside of a complex of enzymes called complex 2 of the electron transport chain, and FAD is covalently attached to the enzyme that's actually doing the work, the succinate dehydrogenase. So it's kind of the connection between the TCA cycle and the electron transport chain. Our next step, which is step 7 for anyone keeping track, is fumarate becomes malate. Now, that fumarate molecule has a carboxylic acid on carbon 1 and carbon 4, and then that double bond between carbons 2 and 3. A reason why the previous step was important is because fumarase isn't called succinase. And what I mean by that is that the enzyme fumarase doesn't recognize this molecule without the double bond where it's at. So fumarate has the double bond, so that's why this enzyme acts on it. And what's going on is the enzyme sort of recognizes that there's an opportunity to make more electronically dense molecule. And so what it's going to do is it's going to add water across the double bond. Now, you may or may not remember this from taking organic chemistry, but this is a pretty standard addition reaction that you'd want to be able to think about or at least visualize for the MCAT. So this water, this H2O, is going to be torn apart, and the OH is going to be attached to carbon 2 or 3 that's participating in this double bond, and that breaks the double bond. Because remember, that second bond there, it's just electrons. So we're going to pair the OH up with one carbon, break the bond, and they're going to be sharing electrons and whatnot, and then the other carbon is going to just get an H. So carbons are still attached to the right number of things, and they're sharing electrons the way they want to, and that's what I mean by adding water across the double bond. Not to harp on orgo too much, but the fun fact here, or the interesting thing here, is rooted in orgo, so we'll dive in a little bit. So because the double bond is planar here, we're going to be able to produce two isomers. We can create an L or a D, malate. If you remember from orgo, L and D are relative configuration, not absolute like the ROS is. And I'm not going to go down the rabbit hole of explaining what all this is. There's an episode out there for that, but that's its fun fact to help you remember how it all works. All right, last step, I promise. Malate becomes oxaloacetate. So this is our final step in the TCA cycle. We regenerate oxaloacetate by squeezing in one more redox reaction, which is stealing the electrons away from the substrate so we can put it into the ETC. We use a molecule called malate dehydrogenase, and we produce NADH. So NADH is the shuttle here. So this last step is really important to have on your radar because it's like an intersection of multiple metabolic pathways. And it turns out, and we are going to talk about this in detail when we get to regulation, but for the time being, it turns out that if glucose is needed in a cell, or if it's primarily like the liver, that if the liver needs to make more glucose to share with the rest of the body, it can take oxaloacetate, it can turn it back into malate, and then malate can jump on this shuttle, it's called the aspartate malate shuttle, and can move from the mitochondria back into the cytosol, where it's reoxidized into oxaloacetate and enters in gluconeogenesis. Alrighty, let's recap. So the overall reaction of the TCA cycle, if you're taking notes or keeping track, is acetyl-coa plus 3NAD plus FAD plus GDP plus 1 inorganic phosphate plus 2 water is going to give us 2 carbon dioxides, a CoAsh, or acetyl-CoA that's a protonated thiol, 3 NADH, 3H plus, FADH2, and a GTP, which is functionally ATP. So I don't think you need to have that memorized, because if you really had to figure that out for a question, which I think would be rare or non-existent on the MCAT, you could. Like if you know the logic and what goes in and what goes out on each step, you could piece that together. So I wouldn't bother memorizing that. But I brought it up so I can make a point. So notice that we have the 3NAD, the FAD, and the GDP on the left side of this sort of equation. They're all energy deficient. We can make NADH, we can make FADH2, we can make GTP, but then we pair it with acetyl-CoA. And I like to think about it as if the acetyl-CoA is a pizza, it's sitting in its delivery box, and the NAD, FAD, GDP are me and my hungry friends. And so when you eat the pizza, you become a higher energy version of yourself, and then you throw out the cardboard delivery box. So cardboard, carbon dioxide. Maybe you see where I'm going with that, but I think that's a useful analogy to remember that we are pairing this rich source of electrons, the acyl group, up with all of these really energy-hungry molecules, and we're just transferring those electrons, or feeding these other hungry molecules, if you will. We'll do a little bit more bookkeeping and accounting, and we're going to talk about this again, probably in more detail for the electron transport chain episode, but I want to talk about how many ATP we're getting per all these processes. So depending on who you talk to, or depending on what article or research that you go and look at, for 1 NADH, we get roughly 2.5 ATP. So the electrons that we get from NADH will help us generate about 2.5 ATP. We have 3 of them in this process, so we get 7.5 per turn of the cycle. We only get 1.5 ATP per FADH2, and that's going to be, I'll explain that more when we get to ETC. And then we get 1 ATP because of that step where we create GTP. You just swap out the guanine for an adenine. So just from the TCA cycle alone, we have enough energy to make roughly 10 ATP per 1 pyruvate, which means for 1 glucose, we get about double that, which I think is kind of cool. Now, regulation is a big deal on the MCAT. That tends to be the kind of questions that you get in biochem, whereas the counting of ATPs and like the structures and chemical reactions that are occurring during these cycles, it tends to be in Chem Phys. But we're going to cover that because it's so important, in a different episode. But something I want to leave you with is to remember that if there's energy in the cell, in the form of ATP usually, like if we got energy, then the TCA cycle isn't really doing anything. Just kind of waiting around because we don't need the energy. But if the energy is low, if you're exercising, then you need to be drawing acetyl-coa into the Krebs cycle so that you can make that ATP. And then again, to connect to the exercise, you're going to be breathing off the carbons that are in acetyl-coa. And this is where we'll leave off for today, at least. Next time, we're going to turn our focus to the electron carriers and the electron transport chain. We're going to talk about the complexes and really give you a good idea about what you need to know for the MCAT. All right, but first we have our quiz. And I've been hearing from some of you that kind of like the idea of the quiz. I have had some suggestions on different types of questions that I'll be working those in for sure. But if you like these, continue to let me know, or if you would like to see them a little bit different. I'm all ears. So question 1, how does a synthase differ from a synthetase enzyme? I would encourage you to think this out. If you need to pause so that you can really collect your thoughts, do so. All right, so both synthases and synthetases create new molecules. That's kind of why this is confusing. But the difference here, the big difference, is to remember that synthases do not directly require ATP or make it. And then synthetases either directly require it or they make it. So in the TCA cycle, step 1 uses citrate synthase, and that adds oxaloacetate to acetyl-CoA, and it creates a citrate. While in step 5, succinyl-CoA synthetase creates GTP, and it utilizes the free energy from the cleavage of that thioester bond. All right, question 2. Why are the thioester linkages connecting coenzyme A to substrates important to the TCA cycle? Kind of already gave this one away, but collect your thoughts, and if you need a second, pause. Thioester linkages are not very stable. So whenever we move from an unstable state, if you will, or configuration, to something that is more stable, well, there's going to be energy that's given off. So the cleavage of the thioester link, and we just discussed the 2 scenarios that are relevant to the Krebs cycle in the last question. When we cleave that, we're going to end up in a much more stable, environmental configuration, and so less reactive, more stable, lower energy. So that energy has to go somewhere, which we're going to harness to make new molecules like GTP. All right, so this last question is more clinical, and I guarantee when you're a doctor, you're going to have this experience. All right, so you're going through, you're seeing new patients, and you walk into this room, and the chief complaint is that they're having a panic attack. Now, of course, you're going to have to address the psychological component and the crisis that's going on there. But first, physiologically, you have to sort them out, because they're complaining of things like, oh, my fingers are tingling, I can't feel my lips, common associated symptoms, and you're like, what's going on? So you want a better picture, so you order a blood panel. Blood panel comes back, and you find out that their blood pH is like 8. And you're like, oh shoot, blood pH should not be that high. I need to figure out how to lower it. And the question is that I want you to think about is like why that's occurring. And this is not only related to the Krebs cycle, it's also connected to other biological processes. I want you to think for a moment, what's going on? Why is their blood pH too high? So take a second, think about it, and then I'll kind of review the case. All right, so your patient, panic attack, hyperventilating, blood pH is too high. So they're alkalotic. We need to acidify their blood back into that Goldilocks zone. So what's happening is that the lungs, the gas exchange, the carbon dioxide out of the body and the oxygen into the body, is not right. Like there's a problem there. You might be thinking, or maybe you have reviewed this idea before, why does that have anything to do with blood pH? So we're going to talk about our very favorite bicarb buffer system, where water and carbon dioxide form to make carbonic acid, and then that carbonic acid becomes bicarbonate, and that bicarbonate is able to resist changes in proton concentration in the blood. But if we're breathing off too much CO2, according to Le Chatelier's principle, we are going to make more of that bicarb turn into carbonic acid and then turn into carbon dioxide and water because we're breathing off the carbon dioxide so fast. So that is how we're making the blood too basic. Now, the reason why this happens in the first place is because the metabolism, in particular the TCA cycle, is not going quick enough. It's not fast enough or enough in our cells to replenish the CO2 that enters into the bicarb buffer system. So metabolism is where all of the CO2 in that system comes from. If you think about exercising, if you're go on a run or something like that, well, you don't hyperventilate and your blood doesn't get all alkalotic and cause all these problems, and it's because your metabolism is keeping pace with that respiration. So that's sort of the answer to that question. Let's look at the reverse scenario though, real quick. I think that might be fun. So again, you're a doctor and you walk into a room and let's make it fun. It's a cop who's there with some issues because they were on this chase after a suspect, and they were running super duper fast and they sort of had to stop because they kind of collapsed or something like that. Oh shoot, this person, they overexerted themselves or something like that. And again, you run blood panels so you can see really what's going on, and they are acidotic. They have way too much protons or acids in their blood. And if you think about it, very, very similar situation. It's a little more complicated, but I think in principle it can illustrate the point. Our metabolism is going really ramped up. And not only are we producing CO2, and we are blowing it off, but more of it is kind of pooling in the blood. We're also producing other acidic byproducts like lactic acid. That's another one that comes from, we talked about that last time, the anaerobic metabolism, which our muscles, especially muscles if you're sprinting with a bulletproof vest and all your stuff on as a cop after a suspect, you are doing a lot of anaerobic metabolism in your larger muscles like your legs and whatnot. So here we've essentially dumped in too much metabolic byproduct, again, primarily from the TCA cycle and a little bit from the lactic acid, and we can't blow off the CO2 fast enough. So that is what would happen in that case. All right, so I will leave us there. Again, next time we are going to talk about the electron transport chain, and we're also going to have a special episode coming up very soon with an individual. His name is Harrison White. He's going to give some advice and weigh in on whether or not you should retake the MCAT. Until next time.
Each episode of MCAT Basics is brought to you by Med School Coach. To access Med School Coach services, including MCAT tutoring and medical school admissions advising, visit our website at medschoolcoach.com. Good luck as you prepare for the MCAT, and we hope you tune in again next time.